All right, everybody, welcome back. You are on week 2 of watching these videos. So I apologize because I know these videos are not very good, and I probably mess up a lot of things, and the sound is weird on the videos. So I understand that, but hang in there with me. You guys are doing amazing. This week will be a lot less videos and more activities and a lab. So you'll kind of have reached that point in this unit where we can do more hands-on. All right, first off, I'm going to have you do a bell quiz. So I need you to turn to the front page of your notes packet, and you're going to put today's date in another box, and really you can just do the box right next to the first one where you put the bell quiz. So put today's date, March 14th, and then you're going to have 6 questions. So don't write too big, because you want to make sure you have room to answer all 6 questions. Okay? All right, so I'm going to read through them, and then we can pause the video and give you a chance to answer these. And once again, you can work with your partner. You are just not allowed to look up the answers in your notes packet. Okay, I really want you to recall, go back, especially where you had a weekend, but go back and recall and remember the different things that you learned. And if you're absent, do the best you can, working with your partner. Hopefully they were here. And you will get full credit as long as you do the quiz, even if you get them wrong. Okay, but answer the questions related to the question. Okay, so number 1, what enzyme proofreads in DNA replication? What enzyme can proofread? Basically, if something gets messed up when they're building it, they can fix it. So what goes along with the proofreading is also the same enzyme that builds it. Okay, number 2, what is the replication fork? Number 3, what enzyme bonds the old and the new strand together? And number 4, what type of bonds are found between the nitrogen bases? And what type of bonds are found between the nucleotides? Okay, we're going to pause this and give you a chance to answer these 4 questions. Once again, work with your partner. Okay, let's move on to the next 2 questions. Okay. Get rid of that. Number 5, what 3 parts make up a nucleotide? So I want you to draw one, and then you can just label what those parts are. Okay, so what parts make up a nucleotide, and draw one. Okay, number 6, what does anti-parallel mean? We talked about this when it came to DNA replication. And what does anti-parallel tell you about the DNA double helix? All right, so same thing. We'll pause this, give you a chance to answer these questions. Okay, I'm going to go ahead and go over the answers. First off, number 1, what enzyme proofreads the DNA replication? So the enzyme that proofreads is also the enzyme that builds it up, and that would be your DNA polymerase. So it's poly the angel because it builds it up, and if there's a problem, it fixes it. Okay, so that's your DNA polymerase. Number 2, what is the replication fork? And this is the area where the DNA double helix splits into 2, and it's the location where you would find the helicase enzyme, breaking apart those hydrogen bonds. Okay, number 3, what enzyme bonds the old and the new strand together? Okay, this one is your ligase. Okay, ligase. All right, number 4, what type of bonds are found between the nitrogen bases? So those are your hydrogen bonds. They're weak. They're in the middle. And then what type of bonds will you find between your nucleotides? And those are your strong bonds, and those are covalent. Okay. All right, number 5. What 3 parts make up a nucleotide? Phosphate, sugar, base. Okay. Hopefully you're able to draw a nice, beautiful picture, right? We've done this several times last week. Phosphate, sugar, and a base. I'm going to go ahead and choose A. Okay. What does anti-parallel mean? All right, so when it comes to anti-parallel, hopefully you remembered that is 2 strands that are complementary to each other, but they move in opposite directions. Okay. So this would be my DNA strand. This would be the 5 prime. And this would be the 3 prime. Okay. So here, once again, you got your phosphate, and then it's made up of your sugar and then you have a base. Right, and this just continues, a phosphate, a sugar, a base, a phosphate, a sugar, a base, right? So we can go ahead and add these other nucleotides on here just visually to get an idea. I know this is really not the best for drawing, but that's all right. Woo, that's a big old covalent bond. All right, so this DNA strand goes from a 5 to a 3. Now, antiparallel, once again, they're complementary strands, but it tells the helicase what direction to travel. So the helicase, I'm sorry, not the helicase, the polymerase, remember Paula the angel builds it up and fixes it. So the polymerase is going to come in, and it's going to bring in a complementary nucleotide. So here comes the complementary nucleotide. There's my sugar attached to it, and there's my phosphate. Okay. So it kind of tells the direction which they go. Now over here, you have the 3 and the 5' side. So it's going to go a different direction because the polymerase always goes from a 5 to 3. So we're going to draw an arrow because it's going to work it this way. So it's going to add a nucleotide. I'm just going to put A here. We'll just know there's a phosphate and a sugar there. It's going to bring in the complementary nucleotide that pairs up to it. So here comes the T. Once again, the sugar. That was bad. And your phosphate. So you have 2 complementary strands, both moving in opposite directions from a 5 to a 3' direction. That was a whole lot. Hopefully you're able to get something written down about what I just talked about as far as antiparallel. All right, so at this point, we are going to go ahead and take a Canvas quiz. So I need everybody to grab their own individual Chromebooks. Okay, once you grab your Chromebook, get into the Biology Canvas page. And once again, here is what we're looking at. Okay. So there's 2 different ways you can get to it. You can click on the schedule. I kind of like to go to the schedule because I like you to see where all this information's at, where you can find out exactly what we're doing day to day. So I'm going to scroll down here to Monday, and there is your DNA quiz, okay, on DNA replication. 10 points. Or, obviously, you can go the quiz route a lot quicker. And that's your quiz. You're taking 3.1 DNA replication. All right, go ahead and work through that. It shouldn't take you more than 5 minutes to do. And when you are finished, please return the Chromebook. Make sure you guys plug them in. Okay, now that the quiz is done, we're going to go ahead and dive into our notes packet. I'm just going to scroll down here a little bit, just kind of going over this. So you should have 2 Bell quiz boxes used up. Okay, one from Friday and one from Monday. And you should have watched this video last week on Wednesday. I did this little fingerprint activity on Tuesday of last week. Okay. Keep rolling. We've did all these notes. Now, the DNA extraction lab and the DNA research, pick a topic. We have not done these yet. All right, that'll be coming up. All right, we talked about our DNA structure. We went through our DNA replication. Here is where we're at, right? So modeling DNA replication. I love this picture because once again, you can see there's your DNA double helix. You have a little circle here. And I'd be thinking, there we go. It's our DNA helicase that breaks apart those hydrogen bonds. It's broken apart. And then these are just free-floating nucleotides that are just floating around inside the nucleus. And you can tell your phosphates are always your round circle. Your sugar is always your penthouse. Okay. 5 sides for the pent. And there's the house. There's your deoxyribose. And then there is a nitrogenous base. So, what is the enzyme that takes these free-floating nucleotides and brings them and hooks them up? Hopefully you remember that that is your DNA polymerase. Now, if you take a look at this, right, you can see this nucleotide, phosphate sugar base. And it's been hooked up. You can see those two lines. Well, those two lines are representing those hydrogen bonds. So we have 2 here. We have 3 here. So we know that these nitrogenous bases have to be A and T, and these nitrogenous bases have to be C and G because of the number of hydrogen bonds, right? Now, what's the enzyme that makes this happen, that bonds them together? And that is your ligase. Okay. All right, so what you're going to do with your partner, I want you to answer question number 1. You're just putting things in order. Number 2, locate the DNA helicase on the DNA model. Answer the question. Number 3, what rule is used to join the free nucleotides to the exposed bases on the DNA? And number 4, I'm going to see if you can work through this and see if you can get an idea of what it's asking to do here. So it says the proportions of the bases are consistent within a species. However, they do vary. I probably should make that a capital, huh? However, they do vary between, oh, now it needs a comma, between species. Using the base pairing rules, complete the following table to show the percentage of each type of base in 5 different organisms. So here's the different species. Okay, you got the human species, and remember A is adenine, and your G is guanine, C is cytosine, and T is thymine. So here's the nitrogenous bases, and in the human species, we have 31% of the DNA sequencing is adenines. So 31% is made up of A's. And 19% is made up of cytosine, the C's. Well, that's all the information you need to fill in what percent would guanine be and what percent would thymine be. So let's see if you can talk this through with your partner, figure out what it's asking you to do there. And then we'll come back and go over the answers. Okay. All right, so you're completing this page, the model DNA replication, with your partner. All right, good luck. All right, let's go ahead and go through these answers. Okay. So we look at the very first one. We're going to number the steps in order to describe the replication of DNA. So let's just find where is step 1. We know in step 1, it's when the helicase comes in and breaks apart the hydrogen bonds. So let's see, hydrogen bonds between nucleotides form. No. Hydrogen bonds between nucleotides break. Strands of the DNA separate. Free nucleotides are attracted to the exposed bases. So the very first step here we're looking at is right here, number 1. Hydrogen bonds between nucleotides break. That is your helicase. So we know we're looking at number 1 here. Okay, number 2, the next step, the strands of the DNA are going to separate. The third step, we have free nucleotides are attracted to the exposed bases on the loose strands of DNA. And once again, they are going to hook up according to the base pairing rules. Okay, A is always going to go with T. And C is always going to go with G. Wow, this pen is really slow to write. It keeps pausing on me. You get the idea. All right, A's with T and C's with G. Okay, so once those nucleotides are attracted and they're next to it, they are ready to bond and create new strands. So this would be number 4. Hydrogen bonds between nucleotides form. All right, so locate the DNA helicase on the DNA model. What we know here is our DNA helicase. Oh, let's do a new color, a little purple pride. All right, so there's our DNA helicase. And what type of biological molecule is a DNA helicase? Well, it ends with ACE. And if it ends with ACE, we know it is an enzyme. Ooh, let's see if I can write this cursive. Then it won't stop me. There you go. Hopefully, oh, I forgot. There's a Y. My cursive. All right, so what type of biological molecule is a DNA helicase? It is an enzyme, which is a protein. Once again, here comes my cursive. Bam! That's a T. All right. Okay, next question. What is the role of the DNA helicase in the replication?